Imagine you are looking through a high-powered microscope at the brain tissue of a patient who recently passed away from a severe neurodegenerative disease.
Right, you're zooming in on those dying neurons.
Exactly. And you immediately see these massive microscopic trash heaps, just, these really dense clumps of misfolded proteins clogging up the inside of the cells.
It's usually the first thing that catches your eye.
Right. And instinctually you think, well, there is the murder weapon. I mean, it seems incredibly obvious, but what if those giant trash heaps aren't the killers at all?
Yeah. What if they're actually the cell's desperate kind of last-ditch effort to save its own life?
It is a wild thought because it completely upends how we traditionally think about cellular pathology. We are so conditioned to look for the most visually obvious anomaly and, just assign blame to it.
We really are. But as we know, molecular biology rarely operates on surface-level logic.
And that counterintuitive reality is exactly what we are dissecting today. We're doing a deep dive into a comprehensive review article from the November 2025 issue of Cell Death and Disease.
Yeah, the paper led by Clevio Nobrega and his colleagues.
That's the one. It essentially maps out the exact mechanisms behind a group of 6 rare inherited brain-wasting conditions known as Polyglutamine Spinocerebellar Ataxias, or PolyQ SCAs for short.
Which is quite a mouthful, but we are talking specifically about SCA1, SCA2, SCA3, which is also known as Machado-Joseph disease, and then SCA6, SCA7, and SCA17.
Okay, let's unpack this. Our mission today is to map out the exact molecular domino effect that takes a single genetic stutter and turns it into devastating neuronal death. But first, why should you, listening right now, care about these 6 specific rare diseases?
Because they provide a literal master blueprint.
Exactly. Understanding the precise step-by-step collapse of a neuron in these diseases gives us a master key for understanding neurodegeneration as a broader category.
Yeah, I mean, the cellular domino effect we're about to explore, it echoes through the pathology of Alzheimer's, Parkinson's, and Huntington's.
So let's get right into the root cause, the primary hallmark, the genetic stutter that starts this entire cascade.
The mechanism here is deeply fascinating. In the DNA of patients with these diseases, there is an abnormal expansion of a specific trinucleotide repeat. Cytosine, adenine, guanine, CAG.
CAG. And because CAG codes for the amino acid glutamine, which is represented by the letter Q, this genetic expansion translates into an abnormally long chain of glutamines.
Right, it creates this long PolyQ tail at the end of the resulting protein.
And just to visualize the physics of this glutamine is a polar amino acid. So when you string too many of them together, you completely alter the thermodynamics of the protein's folding process.
It forces the protein to adopt these really abnormal beta sheet conformations, making it incredibly sticky.
It's a structural disaster. I mean, the protein can no longer fold into its functional three-dimensional shape, right?
Exactly. It misfolds, it exposes these hydrophobic regions that really should be buried safely inside, and it starts aggregating with other proteins.
This makes me think of a manufacturing defect in a highly complex physical gear. Like if a factory accidentally prints a gear with an extra millimeter of metal on one of the teeth, that's our PolyQ tail, it doesn't just stop the gear from turning.
No, it causes active damage.
Right. When you force it into the machine, that extra metal physically shreds the adjacent gears, stripping the entire mechanism.
That's a highly accurate way to look at it. The extra metal causes localized destruction. And this brings us back to that counterintuitive finding you mentioned at the very beginning.
Right. Because if these protein clumps are physically building up in the brain, they must be the murder weapon, right? For decades, the assumption was that those massive visible inclusions were the primary agents of neuronal death.
What's fascinating here is that Nobrega's paper points out that when you actually map the physical location of these massive inclusions in postmortem brains, they just don't align with the regions suffering the most severe neuronal loss.
Wait, really? So the areas with the most cell death don't have the biggest clumps?
Precisely. The regions with the highest density of inclusions often show relatively preserved cell counts. Meanwhile, the regions experiencing catastrophic cell death might have very few large inclusions.
Oh, wow. So the giant aggregates are actually protective reservoirs.
Yeah, this paradigm shift suggests the cell senses these toxic, sticky proteins and actively sequesters them into a centralized trash heap just to get them out of circulation.
I see. So the real danger isn't the trash heap itself, it's the loose garbage.
Yes. The truly toxic agents are the soluble oligomers. Those are the smaller, free-roaming clusters of mutant proteins that haven't been quarantined yet.
Because of their sticky PolyQ tails, these oligomers engage in extensive toxic gain-of-function interactions, right?
Exactly. They don't just sit there. The paper highlights SCA1 as a prime example of this. The mutant ATXN1 protein actively binds to critical transcription factors and just pulls them out of circulation.
It literally drags essential genetic machinery into the aggregates, which leaves the cell completely unable to transcribe the genes it desperately needs to survive.
But, the cell isn't entirely defenseless. It attempts to mitigate this by modifying the proteins after they are translated.
Right, post-translational modifications, or PTMs.
Right.
Like phosphorylation or S-methylation.
Yeah, PTMs are basically the cell's way of dynamically regulating protein function by adding chemical tags.
I was reading the section on how these PTMs get hijacked, and the contrast between the different diseases is just wild to me. I assumed phosphorylation would have a uniform effect, but it is incredibly context-dependent.
It is so specific. Like, look at the biochemical difference between SCA1 and SCA2. In SCA1, if the cell phosphorylates the mutant ATXN1 protein at a specific serine residue S776, it actually changes the local charge.
And that prevents the cell's ubiquitin ligases from recognizing it, doesn't it?
Exactly. The phosphorylation stabilizes the toxic protein, it shields it from degradation, and massively exacerbates the disease.
It's essentially putting a bulletproof vest on the rogue oligomers.
Yeah.
But then in SCA2, phosphorylating the mutant ATXN2 protein does the exact opposite. It flags the protein for destruction.
Which is mind-blowing. The exact same biochemical tool is neuroprotective in one context and highly destructive in another.
It really highlights the immense difficulty in developing broad-spectrum therapies for neurodegeneration. You can't just blindly inhibit or promote phosphorylation across the board.
No, you have to understand the precise structural mechanics of the specific mutant protein you're dealing with.
Speaking of mechanics, here's where it gets really interesting. Let's talk about the RNA, because this completely broke my understanding of basic biology.
Oh, the RNA toxicity is fascinating.
Right. We usually think of the mutant protein as the sole threat. The DNA is the blueprint, the RNA is just the passive messenger, and the protein is the physical weapon. But this review heavily emphasizes RNA-mediated dysfunction.
It's a dual threat mechanism. Those expanded CAG repeats in the RNA aren't just passive sequences of letters. Due to their length and thermodynamic properties, they fold back on themselves,
forming those incredibly stable, abnormal hairpin structures.
Yeah, and these hairpins aggregate into what we call RNA foci.
And just like the protein inclusions kidnap transcription factors, these RNA foci trap essential RNA-binding proteins, preventing them from regulating normal cellular splicing.
But the RNA toxicity goes even further than that.
Yeah, I want to dig into RAND translation, because reading about this felt like reading science fiction. Normally, to translate RNA into a protein, the ribosome absolutely needs a start codon and AUG sequence.
Right, this classic start signal.
But the source indicates these long CAG repeats just bypass that rule entirely. How is that mechanically possible?
Well, RAND translation, which stands for repeat-associated non-AUG translation, is basically a mechanical hacking of the ribosome.
A mechanical hacking.
Yeah. The current model suggests that the abnormal secondary structure of the RNA, that tight hairpin we just talked about, it physically stalls the scanning ribosome.
So the sheer physical roadblock of the hairpin forces the translation machinery to kind of slip and begin translating without the AUG methionine start codon. So the cell's factory line hits a structural speed bump, glitches, and just starts blindly printing out whatever code is in front of it.
That is exactly what happens, and because it's slipping, it translates across multiple reading frames.
Wow.
So from a single expanded CAG sequence, the cell is suddenly churning out multiple different types of highly toxic, aggregation-prone peptides, all without a standard start signal.
It completely subverts the central dogma of molecular biology. Okay, so if we take a step back and look at the cellular environment right now, we have sticky mutant proteins, kidnapping transcription factors, hijacked chemical tags, shielding the toxic oligomers.
RNA hairpins trapping splicing proteins.
And ribosomes glitching out to produce rogue peptides. When a neuron is facing that level of internal sabotage, what is the first major cellular system to buckle under the pressure?
The metabolic engine. The mitochondria take a massive hit.
Because neurons have huge energy demands, right?
Huge. Especially to maintain their resting membrane potentials and drive synaptic transmission. But these toxic PolyQ oligomers physically interact with the mitochondrial outer membrane and disrupt the electron transport chain.
Specifically compromising complex I and complex II, according to the paper. which means a catastrophic drop in ATP production.
Yes, but the loss of energy is really only half the problem here. When the electron transport chain is compromised, electrons leak out and prematurely react with oxygen,
triggering a massive spike in reactive oxygen species, or ROS. To build on our earlier gear analogy, it's not just that the factory loses power.
It's worse than that.
Yeah, it's like the jammed gears in a nuclear reactor's cooling system are causing localized meltdowns. The reactive oxygen species are essentially molecular fires tearing through the cell, oxidizing lipids, mutating DNA, and just destroying structures.
And normally the cell would deploy its degradation systems to clear out those damaged organelles and misfolded proteins.
The ubiquitin proteasome system, or UPS, handles the individual proteins, right? And autophagy clears out the larger aggregates and damaged mitochondria.
Right, but in PolyQ SCAs, both of these cleanup crews fail completely.
Now, I understand why the sheer volume of garbage might overwhelm the system, but the paper suggests it's an actual mechanical failure. Why can't the proteasome just chew up the mutant proteins?
It comes down to physical geometry. The eukaryotic proteasome is essentially a narrow barrel. To be degraded, a protein must be unfolded and threaded through the tiny pore of the 20S core particle.
But the long PolyQ tract physically cannot be unfolded properly.
Exactly. It forms a structure that is highly resistant to being pulled apart, so it gets stuck. It's literally like trying to pull a thick knot in a thread through the eye of a needle.
It jams the entry pore, completely disabling that specific proteasome. But what about autophagy? If the trash compactor is jammed, why not just bag up the whole mess and send it to the lysosome?
Because the logistics network itself gets severed. Well, for autophagy to work, cargo receptors, like the protein P62, have to bind to the toxic aggregates and escort them to the autophagosome.
Okay.
But in these diseases, P62 and other crucial clearance proteins get physically sucked into the aggregates and permanently trapped there.
Oh, man. So the cell has the machinery to destroy the garbage, but the escort proteins are locked inside the burning building.
That's exactly it.
So the neuron is starving for ATP, it's burning from oxidative stress, and it's overflowing with toxic waste. That brings us to the secondary hallmarks, that cascades affecting how the cell actually functions within a neural network.
Right, because neurons don't exist in a vacuum. They exist to transmit signals.
If a cell is in this much metabolic distress, how does that alter its internal wiring and epigenetics?
We see profound transcriptional dysregulation, and this is largely driven by the altered balance of histone acetylation.
Remind me, DNA is normally wrapped around spool-like proteins called histones, right?
Yes. When the cell needs to read a survival gene, histone acetyltransferases add a chemical tag that relaxes the chromatin, allowing transcription.
But those mutant PolyQ proteins, especially in SCA1, 3, 7, and 17, they accumulate in the nucleus and sequester those acetyltransferases.
Leaving the deacetylases to run completely unchecked, the chromatin condenses into tight heterochromatin.
So the DNA is wound so tightly that the transcription machinery literally cannot access it.
The genetic instructions for synaptic plasticity, structural repair, and basic survival are essentially locked in a vault.
If the cell can't read its own repair manuals, its signaling is going to misfire. And this leads us to one of the most critical mechanical breakdowns in the entire paper, the hypervulnerability of Purkinje cells in the cerebellum.
This is such an important point.
We know the cerebellum coordinates our physical movements, which is why ataxia, that loss of muscle coordination, is the defining symptom of these diseases. But why are Purkinje cells specifically in the crosshairs?
Purkinje cells are incredibly unique. They are among the largest neurons in the human brain. They feature these massive, highly complex dendritic arbors that process thousands of incoming signals simultaneously.
And to manage that immense signaling load, they rely on exquisitely precise, tightly controlled fluctuations of intracellular calcium.
Yeah, calcium isn't just a mineral here. It's the primary messenger dictating exactly when and how the cell fires.
And the mutant proteins directly sabotage that calcium regulation, don't they?
They do. Mutant proteins like ATXN2 and ATXN3 physically interact with IP3 receptors, which sit on the membrane of the smooth endoplasmic reticulum inside the cell.
And the endoplasmic reticulum is basically the cell's internal calcium vault.
Right. The mutant proteins bind to the cytosolic domain of the IP3 receptor and hypersensitize it.
So they essentially tick the lock on the vault.
Yes. They pop the floodgates open, causing a continuous leak of calcium from the internal stores into the cytoplasm.
And because Purkinje cells are so hypersensitive to calcium concentrations, this chronic flood acts as an excitotoxic poison.
The elevated calcium forces the neuron to continuously fire, while also activating destructive enzymes like calpains that physically tear the cytoskeleton apart.
The neuron literally fires itself to death. That is deeply tragic at a cellular level. The cell is desperately trying to do its job, but its internal messaging has been hijacked to force it into overdrive,
Which pushes the cell past the point of no return.
moving us into the end-stage hallmarks, the final toll.
If we connect this to the bigger picture, this is where the local tragedy becomes a systemic crisis. The continuous death of these massive Purkinje cells triggers the end-stage hallmark, severe neuroinflammation.
The brain's resident immune cells, the microglia and astrocytes, activate in response to the damage. Now, you would think an immune response is a good thing, right? Like the paramedics are arriving at the scene to clear the debris and protect the surviving tissue.
But the environment is just too toxic. Instead of remaining in a supportive role, the microglia undergo a morphological shift into a highly aggressive pro-inflammatory state.
They start releasing cytokines like TNF-alpha and interleukins.
Yeah, which are meant to clear out infections but end up severely damaging the already vulnerable surviving neurons.
The review paper makes a fascinating point here. It notes that gliosis and astrocyte hypertrophy, these massive immune responses, are actually observed in mouse models before clinical symptoms or widespread cell death even appear.
It's a crucial detail.
That blew my mind. The inflammation isn't just a reaction to the end-stage destruction. It is an active early participant in driving the cells to die.
The immune system essentially creates a hostile microenvironment that accelerates the pathology. It's a vicious feed-forward loop.
The struggling neurons release distress signals. The microglia overreact and release toxic cytokines, which puts more stress on the neurons,
Causing more mutant proteins to misfold, and the cycle continues until the tissue is completely decimated.
which forces us to ask the ultimate question of selective vulnerability. If this is a genetic disease, every single cell in that patient's body, from a skin cell to a liver cell to a prefrontal cortex neuron, has the exact same CAG stutter in its DNA.
They are all producing the mutant protein.
So why do the Purkinje cells completely collapse, while a skin cell just hums along just fine?
Selective vulnerability is really the grand puzzle of neurodegeneration. Based on the mechanical breakdown we've just traced, the review points to a perfect storm theory.
Oh, okay, a perfect storm.
Yeah, a skin cell can tolerate the mutant protein because it doesn't have the immense ATP demands of a massive dendritic arbor. It doesn't rely on razor-thin margins of intracellular calcium to function.
And it might not even express these specific transcription factors that the mutant ATXN1 protein likes to kidnap.
Exactly. It's the specific intersection of the cell's baseline environment and the mutant protein's toxic gain of function.
The Purkinje cell requires massive energy, which the mutant protein denies by damaging the mitochondria.
It requires precise calcium, which the mutant protein floods by jamming the IP3 receptors open.
It requires specific genetic pathways, which the mutant protein locks away by altering histone acetylation. The Purkinje cell isn't just damaged, its specific physiological identity is weaponized against it.
It is a devastating cascade when you look at it all together.
So what does this all mean? If we look back at the entire map of this catastrophe, the interconnectedness of it all is just staggering. We start with a primary genetic stutter that alters the physical thermodynamics of a protein and twists RNA into toxic hairpins.
Right.
Those primary culprits then trigger the secondary cascades. They jam the proteasome barrels, spark oxidative fires in the mitochondria, lock away the DNA, and flood the cell with excitotoxic calcium.
Which ultimately provokes that end-stage hyper-aggressive immune response that finishes off the most physiologically vulnerable cells in the brain.
When you lay out the mechanics like that, the difficulty of treating these diseases becomes glaringly obvious. You cannot just design a drug to break up the protein inclusions.
No, because by the time those large aggregates form, the mitochondria are failing, the calcium stores are leaking, and the immune system is already hostile.
If you clear the protein but ignore the RNA toxicity and the calcium flood, the neuron still dies. And that mechanical reality brings us to a final, provocative thought for you to consider.
Yeah, this is the part that really sticks with you.
Throughout this entire discussion, the most startling revelation was RAND translation. The fact that mutant RNA can physically hack the ribosome to bypass the start codon entirely challenges the most foundational rules of biology we were all taught.
It implies that RNA is not just a passive carrier of genetic information. In the context of disease, its physical topography makes it an active, dangerous saboteur.
And if the RNA is the saboteur, if it's a thing hacking the factory before the toxic protein is even fully built, it changes the entire landscape of therapeutic intervention.
It really does.
If we want to truly cure neurodegeneration, Maybe we have to stop chasing the toxic proteins and the jammed proteasomes at the end of the line. Maybe the future of neurology requires us to invent therapies that can safely intercept and rewrite rogue RNA transcripts in a living, breathing human brain before that very first domino even has a chance to fall.
What would a drug that edits RNA in vivo actually look like?
Right. It sounds like pure science fiction, but tracing this mechanical cascade from start to finish, it feels like the only logical next step. Thank you for joining us on this deep dive. Keep questioning the obvious, keep exploring the mechanics behind the mystery, and as always, stay insanely curious.